Okay, now let's see if we can apply some of the stuff that we've learned from Chapter 3. So, thinking about the names of the cellular arrangements that we learned, what do you guys think that each of these would be called? Let's start with this one first. So, those actually are circular bacteria, and two of them are lumped together. We actually see in certain plots too, another two are lined up together. So, based on our terminology, we'd probably call these diplococcai. But you could get a little bit more specific and actually refer to them as tetrads if you'd like. Okay, let's start with, or continue with this one. So these are circular bacteria, and notice that they are in cluster arrangements. So those would be then Staphylococcus. And then looking at these ones down here, we see that those are rod-shaped bacteria and they are in chains. So those would be Strepto, and rods are Bacilli. And then here we have chains again, albeit they're not as straight and nice as these rods, but they are chains. So we do see some chains. So those would be best described as Streptococci. All right, next question. Which of the following structures are possessed by some but not all prokaryotes? Well, we know that all of them have to make proteins, so all of them will have ribosomes. All cells are defined by a plasma membrane, which is also known as a cell membrane, so all of them will have that. All cells have DNA, so our prokaryotic cells either have one or more chromosomes, so that's correct. So flagella. So we actually see that flagella is only in roughly 50% of the bacteria that have been classified thus far. So then that is in some, but not all of our prokaryotic cells. So that would be our answer. Which of the following bacterial appendages is not used for attachment? So when we talked about those different external structures, we talked about how fimbriae were actually helpful for sticking to mucous membranes. Pilus actually allowed for connection of cells. Flagella were more about movements, and slime layer was definitely attachment. Now, when we think about attachment, a lot of times people think about attachment in terms of attaching to surfaces, but if you remember, the pilus also allows for the cells to attach to other cells, so technically that falls into attachment. So our correct answer then would be flagella, because that is for motility and not for attachment. Okay, next question. True or false? The outer membrane contributes an extra barrier in gram-positive bacteria that makes them impervious to some antimicrobial chemicals. So they are generally more difficult to inhibit or kill than are gram-negative bacteria. So if you think about it, the outer membrane, that wasn't associated with gram-positive bacteria, that was associated with gram-negative bacteria. So this is in fact a false statement. We could make it true if we just interchanged the gram-positive and gram-negative words in this statement. Where in a prokaryotic cell would you find the genetic material? So definitely not nucleus, because that's in our eukaryotic cells. Oh, nucleoid? Heck yes it is. So that is the area where we find that DNA. Which statement best describes what happens when a gram-positive bacterium is placed in a solution of distilled water and penicillin? So remember when we talked about our gram-positive bacteria, one of the cons of our gram-positive bacteria is they are more sensitive to antibiotics that will actually affect peptidoglycan synthesis, and penicillin is one of those. So if we take our gram-positive bacteria and we have this thick layer of peptidoglycan, everything's nice and neat, put them in distilled water, well, some water is going to get in there, but nothing will really happen because they have that nice thick layer of peptidoglycan. Now, throw in some penicillin and guess what? We start erasing that outer membrane, and now we have water rushing in with no structural integrity reinforcement with that cell wall. So we would actually see the cell basically having so much water come in that it will rupture, and that is referred to as osmotic lysis. So via osmosis, water rushes in and then causes the cell to rupture. Okay, you have isolated a motile gram-positive cell with no visible nucleus. You can assume this cell has what? Well, ribosomes. We're going to find ribosomes in all cells because they're associated with protein synthesis. So that is a viable candidate. Mitochondria, those are associated with eukaryotic cells. And we just saw that this is a gram-positive cell with no visible nucleus. So if there's no nucleus, it is definitely not a eukaryotic cell, and it definitely won't have mitochondria. Inclusions, we do know that inclusions are associated with prokaryotic cells, which is obviously what we have here since it's gram-positive and has no nucleus. However, I told you all that inclusions can be found in some cells, but not necessarily all cells. So technically, we don't know. It could, but we don't know for sure. So we would cross that one off the list as well. And then our outer membrane is only associated with our gram negatives. We're working with a gram positive, so that's definitely wrong. And since we were able to cross out 3 of these, we know that it can't be all of the above. So our answer then is ribosomes. So all cells possess a plasma membrane, cytoplasm, and DNA, and ribosomes. With that, we conclude our Chapter 3 discussion. So if you have any questions on any of the material in Chapter 3 or any of the questions that we just went over, please do not hesitate to reach out and ask.